Hello, Lana. Welcome back to our studies. Wilfred Momanyi Onkangi-Kiongozi, our YouTube channel. And due to public demand, so many students have written to my comment section in our YouTube channel requesting that I do chapter 3 form 1 work, which is the cell. Today, I'll do quite a number of videos. And please concentrate, have somewhere to write, and follow me through as you share this link to all your networks. With a lot of regards, the students that requested this chapter, led by one from Gara Girls, Form 1 student, stay tuned. In this first video, we shall do a few objectives, and the first one is we shall define a cell. Living organisms are made up of cells, and very many of them. So a cell is the smallest unit. So a cell, this is the smallest unit, smallest, basic stroke, fundamental unit of an organism. So an organism is made up of cells, and this smallest unit, which is basic or fundamental, is what we call a cell. So cells make up tissues. Now this is the organization of an organism, how an organism is maybe constructed or made. From cells we come up with tissues. Tissues make up organs. Organs give what we call organisms. Of course, at the middle between organs and organisms, we have what we call organ systems. So these cells are of different types. We have plants and of course animal cells. So before the cells, because inside the cells, I want to give you full organization. Inside these cells, we have what we call organelles. So we have organelles that are found in cells. Then several cells will form what we call a tissue. Then several tissues will form organs. And I hope I will give you, when we reach the cell specialization chapter, at the end of this chapter, this topic, we shall be talking about types of organs, types of tissues, types of cells, and of course, types of organ systems. That is when we go towards the tail end of this chapter. So the cell is the smallest. I think there should be a comma here. Basic stroke fundamental unit of an organism. If there is no cell, then probably we will not have an organism. This was objective number 1. Number 2, I want us to talk about an apparatus called a microscope. A microscope. This one is an apparatus. Or tools used, can say tool, not tools, used in magnifying. We can also say stroke enlarging cells and structures within the cells. Enlarging cells and structures of cells. I have already told you that inside the cell we have what we call organelles. We shall look at them. But now, cells are very small. We said actually the smallest. So when they are small, we refer to the cells as microscopic. That means they are small. Actually, this one means they are very small. This means they are minute. Due to the fact that the cells are very small or minute, we use some tool which we are calling the microscope to at least magnify or enlarge these cells so that you can be able to do a nice study, or you can be able to see the structures or the organelles or even the cells properly. So for that matter, then cells are referred to as microscopic. When you hear cells are microscopic, that means they are minute or they are very small. You can't see them. You cannot see them. You cannot see them using naked eyes. Naked eyes. So you must use an apparatus we call the microscope. So we shall have two types of microscopes. We have 1 and 2. We have a light microscope. Then we have an electron microscope. Some student will say an electric. No, we say electron microscope, of course, and a light microscope. So as the names state, a light microscope uses light to illuminate your structures or cells. Then an electron microscope uses a beam of electrons. Now, we shall base ourselves more importantly on a light microscope because it is the most common and the one that is examined most of the time. So our next thing, as I open up another page, is to teach you about a light microscope. So a light microscope. A light microscope is an apparatus that is used or uses light to illuminate the specimens. Uses light to illuminate. The source of light can be natural light from the sun. It can also be light from a bulb, because we have some microscopes that you connect to the power or maybe electricity and then you use them, but it's a light microscope. Don't call that one an electron microscope. There is a video in this collection of Form 1 work that I did where I drew a chart. That chart was in yellow. You will find this video here where I have a light microscope drawn and all the parts well illustrated. And in that video, I remember there was a microscope itself. I showed you the mic, so I will refer you to that video. But one thing that is important in this lesson before I wind up this video, we want to look at the parts once again. Parts of a light microscope. Parts of a light microscope, and of course functions of these parts. Most importantly, functions. So the functions of parts of a light microscope. I still refer you to that chart. I was in a red shirt. You will find this video in my collection. So before that, I think I have to show you that microscope. Here it is. The microscope, we have the eyepiece. Here it is, the eyepiece. Then we have the body tube here. We have this bigger knob called the coarse adjustment knob. We have the smaller one, the fine adjustment knob. We have the arm. We have the revolving nose piece, this part here. that revolves, and then below it we have the objective lenses. The two of them here. Although in that video that I'm connecting with this one, we had a microscope that carries 3. Then we have the flat platform stage. This one here, a flat platform where you place the the specimen. Then we have clips, these ones that are on that flat platform. We have the condenser down here. We also have the diaphragm below it. Condenser, diaphragm below it. Then we have the mirror, and of course we have the base stand, and my microscope rests on a given bench. So I want to discuss the parts and functions of these parts one at a time, and then of course we come to the end of this microscope. But still, most importantly, there is a video that I made, a chart. You will find it within the playlist of Form 1 biology work. So let us start with the eyepiece. The eyepiece, part number 1. Part number 1, the eyepiece. The eyepiece has a lens. The eyepiece contains a lens. Contains a lens that works in combination with other lens, and the other lens are objective lenses. So with other lenses, objective lenses, for example, to magnify stroke enlarge specimens structures. So the eyepiece, as usual, has a lens inside it that works in combination with other lens to bring the desired magnification. Number 2, we have the second one we will talk about is the arm. The arm. I'm just choosing the parts randomly. The arm is also called the limb. The arm is also called the limb, and this one is for support. This one supports the body tube. Supports the body tube and stage. Supports the body tube and stage. When I showed you the diagram, I will still show you that this body tube and this stage down there are supported by this arm. So it supports the body tube and the stage. So the work of that arm is for support. We also have the third part. They say another support part. We call it the base. Base, also called the stand. Base, also called the stand. The base stand is to provide firm support. Provide firm support. Provide firm support and stable, actually firm or stable. You can say stable here. Stable support to the whole microscope. To the whole microscope. So when a microscope rests on the bench, like this one here, all the weight of all these parts rests on this base stand. So the base stand is used to provide firm support to the whole microscope, and that's what we are calling the base. The stable support to the whole microscope. Number 3, coarse. Number 4, coarse adjustment knob. Coarse adjustment knob. The bigger knobs. This one is used in raising and lowering the body tube. Let me show you that one. This big one here. Coarse adjustment knob used in raising, lowering, stroke lowering the body tube a longer distance to bring rough focus. So what happens is the body tube is raised. This body tube here, you know, raising it means it goes up like that. Lowering it means it comes closer to the stage. So the work of moving it a longer distance up and down is the work of the coarse adjustment knob. And what happens is it brings what we call rough focus. Not sharp focus, but rough focus. The next one is the fine adjustment knob, the smaller one. Fine adjustment knob, number 5. Fine adjustment knob. This one now also is used for raising, come lowering the body tube, a shorter distance. Now that's the difference. A shorter distance to bring the image into sharp focus. Shorter distance, and also brings the image into sharp focus. In this case, you are now able to see the details of the structures that you are examining. We also have more parts. We go to number 6. We can say the objective lenses. Number 6, objective lenses. Objective lenses, they also have lens. I can show you them here in this small diagram here. They have lenses, the objective lenses, the two here. They have lenses that are also used in magnification, but they work in combination with the eyepiece lens. So this is the second set of lenses. Used in combination with the objective lenses, that is the eyepiece lenses, which contributes to magnification of the specimen, to magnification. I want to repeat that the objective lenses don't work alone. They work in combination with eyepiece lens. So the eyepiece lens together with the objective lenses brings about what we call desired magnification. We are still moving. We will open another page and talk about something like the revolving nose piece. Revolving, this is number 7. Revolving nose piece. This one is another part, and it holds the objective lenses and holds the objective lenses, in position, so that they can be used in, also enabling them again, because it revolves, this revolving nose piece enables change, holds the objective lenses and enables change from one objective lens to another, and that's why we are saying it revolves. It revolves. That means it enables one to change from one objective lens to another. We have a low power objective lens, low power objective lens. We have middle power objective lens, objective lens, and of course we have high power. If there are three, high power objective lens. Depending on the type or the one that you want to use, you will revolve them. And I said there is always when you rotate, when you revolve it, there is always clicking into position. Clicking into position. You will feel, you will hear the sound, the clicking sound. That means you have changed into that objective lens that you are interested in. Then we go to another part. The other part is the diaphragm. Can talk about number 8, the diaphragm. Now I'm using capital letters. Diaphragm. The diaphragm is an aperture, is an opening. Diaphragm is an aperture, aperture. An aperture is an opening, but still can be closed. That regulates the amount of light, passing to illuminate the specimen. So this is an opening. An opening is called an aperture. So it can be opened fully, it can be closed a little, just to regulate. Yeah, to regulate. So if the amount of light is too much, you can reduce that hole or that opening. If it is too little, you can enlarge it. So what happens is this light does not go direct to the specimen. It passes through some point we call number 9, condenser. So a condenser concentrates, CC, concentrates, you see CC, condenser concentrates, but the diaphragm regulates, DR. That's a small formula for those who are interested. Condenser concentrates. To concentrate is to focus. Yeah, to make sure that the light is concentrated. Concentrates or focuses light onto the stage. Onto the stage. So when this light has been focused or concentrated, then that light can be used effectively in magnifying or illuminating the specimen. We have the stage. We have the stage number 10. We have the stage. The stage is a flat platform. The stage is a flat platform onto which the specimen on the slide is placed. So flat platform. And on this stage we have what we call the clips. Of course, a pair of clips that holds the specimen on the slide in position during examination. When we say examination, we mean study. Biological examinations are also called biological studies. So we move to something else. I think we have just very few of them. We have the mirror. Number 12, we have the mirror. A mirror is used to reflect light from the environment that is, or surrounding that is used in illumination of the specimen. Illumination of the specimen. So I think after talking about those parts, I will only emphasize that we have two parts that are very, very common and the difficult ones for a number of students, and that is the condenser and the diaphragm. These ones are very confusing when they are set in the exam. And in my video where I was showing you a chart and the microscope itself, I may have forgotten to emphasize on the two. So the condenser and the diaphragm are the most difficult parts for the students, but we have said clearly that the diaphragm is used as an aperture or an opening that regulates the amount of light. So DR, D, diaphragm for regulation, R. Then C, condenser, concentrates or focuses. C, that is condenser, concentrates lights onto the stage. So till our next lesson, thank you very much for your support. Continue following me. The next video shall exactly be about the precautions while using a light microscope and how we are supposed to handle it while we are using it. That is going to be our next video. See you and keep following me.